Okay, let's give this a go. I'm Kate Scow. This is Soil Science 111, and this is going to be my first attempt at videotaping one of my lectures for the course. So we're starting with Lecture 1. Lecture 1's an overview of the players, all the organisms that are present in soil. First, give a big picture of the food web, and then go in and get an introduction and a general overview of some of the major groups of organisms in the soil. The second part of this lecture will be looking at the different types of interactions among members of the soil food web. Okay, the soil food web, this picture shows you a very simplified version of what is really quite a bit more complicated. There's a lot of arrows that aren't shown there, particularly the arrows that bring the organisms back into the soil organic matter when they die, which is an important part of any cycle. But here you can see that the primary producers are plants. It's interesting, the plants we often think about as being above ground. Of course, they've got to be above ground to have access to the sunlight and be able to capture the energy there through photosynthesis. But they have an equal part of them below ground and where all the other major members of the soil food web exist. So the energy flows through the plant and then moves into the soil, pumped below ground via root exudates, which we'll learn more about later, and also through the decomposition of the plant roots that are there when they die, and then also some of the above ground portion of the plant will fall into the soil and be perhaps tilled or biotilled into the soil. So the front line of organisms that deal with that organic matter are what we call shredders, which are actually larger organisms known as fauna. Fauna are the animals in soil. They're going to break down these big pieces of organic matter that are really difficult for organisms like fungi and bacteria to do too much damage with, because the pieces of organic material are too large. And what you need to do is decrease, actually increase the surface to volume ratio of that material, so that then you have smaller bits that bacteria, archaea, fungi can colonize and help break down. So some of these larger organisms are very important in that, and creating feces, which is also another really important part of food webs, which is really modified organic matter. It's partly digested organic matter that is then released into the soil, ready for other organisms to use. And also, it's inoculated. Feces is inoculated organic matter. Think of an earthworm and all the microorganisms in their guts that are going to be released with the soil organic matter. All right, then the next organisms involved, not that they're not involved earlier on as well, but to a lesser degree, are the fungi and bacteria, and what we don't see there are archaea that are then going to be breaking down this complex organic matter into simpler forms that plants can take up. And then we've got the organisms that are in the higher trophic levels that prey upon the bacteria and the fungi and on some of the other fauna that also are directly degrading some of the organic matter, and then those that prey upon those and those that prey upon those as you move up the food chain. We don't have a charismatic bald eagle here like we do in like a lake system food web. Our charismatic megafauna, I guess, are things like moles and aardvarks and armadillos. Anyhow, so we need to look at how do we classify organisms, and I think you all are well aware of now the old world of the five kingdoms no longer is the paradigm that we use to describe all life forms, and in fact, the kingdoms were based on phenotypic information, so that's information about, the actual attributes of organisms, what is like manifested. And this has led to years and years and years of confusion and shifting, particularly among the microorganisms, what group goes in which category, and also it didn't provide a lot of information that allows you to look at relationships among these different life forms from perhaps an evolutionary point of view. So what we now use, the paradigm that we now use to describe and classify life, is phylogenetics, and phylogenetics is a system based on genetic variation, and then looking at relationships of organisms based on their similarity genetically. And Carl Woese, the fellow in the bottom of the picture, is the person who proposed using a particular gene that is shared by all organisms and that has some interesting aspects that makes it a good candidate, and to use the sequence of that gene to classify life forms. Since all God's children have small subunit ribosomal genes, SSU are RNA. You may remember from biology it's 16S in prokaryotes and 18S in eukaryotes. And the reason this is a good gene to look at, of course there's tons of other genes that could be used, but this is the one that was presented and people tend to agree with. It's not perfect, but it's pretty good. So the small subunit ribosome is possessed by all organisms. It has a high degree of conserved regions. You can see from the pictures below, there are the secondary structures. The ribosomes for bacteria, archaea, and eukarya. And you can see if you just glance at those pictures, there's a lot of similarities among them, even though these are very different groups of organisms. And that's because big portions of these genes are conserved, which is great because then you can line up the genes, the sequences along the conserved regions, but then take advantage of the fact that there are also variable regions, parts of the ribosomal gene that have been subject to short-term mutations. And these mutations have led to differences that then allow us to differentiate organisms. And in general, over time, there's been more and more mutation in these variable regions, and this has led to, we can track this in looking at the evolution of life forms. I'm giving you a very simplified view of this. It's much more complex than how I'm describing it, but the basic principles are there. And this is easy to sequence. And now, using this to be able to compare organisms, we have this very objective, quantitative way to be able to determine relationships among life forms. And here's an example. Here is just a small portion of the small subunit ribosome for a number of different organisms, including humans, but also including a whole bunch of different microorganisms below it. And you can see I put boxes around the conserved regions. Those are the places where the sequences are exactly the same. And then you can see these other areas that are much more variable. And these relationships, these similarities then are what are used to create this map, this universal phylogenetic tree. And the big surprise that came out of doing this was that there were no longer just prokaryotes and eukaryotes. There were in fact 3 domains. There was greater diversity within what we called prokaryote, certainly than there was within eukaryotes. So now we have these 3 domains, larger than kingdom, the bacteria, the archaea, and the eukarya. Now, among the eukarya, you can still see the kingdoms. There's the plants, the animals, fungi. How humiliating. We all are almost identical We know we're not identical because this is only based on the small subunit ribosome, but looking at that gene, there's a lot of similarities among those three very different groups of organisms. And then you can see tremendous diversity in bacteria and archaea. And this tree is a very simplified one. It just shows you a few of the major groups. Also reminding you that mitochondria and chloroplasts are former bacteria that have become organelles inside eukaryotes through endosymbiotic evolution. I think that's what it's called. And you can see that you can map them out with the bacteria and see who their ancestors were. The archaea are an interesting group that were formerly thought of as being only in extreme environments, but we'll learn a little bit more about how important they are in soil as well. I think another interesting thing to see is what we used to call, one group, the protozoa, which is shown here on this figure with ciliates and amoeba, and I think the euglena are scattered throughout sort of a swath on this in this branch that is in the eukaryotes. This website link below, eTree of Life, is a great place, a great website if you want to look at what's going on with this. And of course, this is always changing as well as we get more information. Okay, so here's another picture of that same tree. Do you see the bacteria, the archaea, and the eukarya? And what this has opened up is just an explosion of capability in looking at microorganisms in nature. This has been just a wonderful event for microbial ecologists. So just to back up a little bit, when you find a new organism, what you can do is sequence this small subunit ribosome, and then you can submit, and you can do that sequence anywhere. You can sequence anything you want here on campus at the Genome Center, and you get back the sequence pretty fast. And then you can submit that to a database. The RDP database at Michigan State is one where then your sequence will be matched against existing sequences, and perhaps you will score, you'll find, whoa, it's a Thermoproteus, blah, 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 and you get a really good match. Or you may find, like with these other sequences here that just have numbers and initials, that there isn't a match in the existing database, and but you can get a ballpark of where your organism falls and who it's related to, which gives you a lot of gives you a good head start in thinking about what that organism might do. The other great thing about this then is, because all these data exists, there's all these molecular tools that you can make based on these known sequences. And we'll talk about this later on in the class, but probes and primer sets can be developed based on these known sequences, and then you can take these tools and go and look for organisms in nature. So you can have like a probe for that Thermoproteus if you want to go then and go fishing for it in your soil sample and see, do you have any Thermoproteuses in your sample based on the match to these known sequences. Now, the weakness of this approach, it's not a weakness, but it's just a limitation of it, is that there are a lot of organisms that have not been identified yet. So, and it's good people keep finding new organisms and submitting them to the database. It's required to put them on a database if you want to publish anything about a new organism. But you may not have existing sequences to even know about the existence of another organism. And for example, is like if you want to go look for some Pseudomonas, let's say anthrax, let's be more exciting. and see if you have that in your backyard. And because anthrax does live in the soil, you may have a sequence for one particular kind of anthrax and not for another kind. So you may not be able to look for the organism that you might actually want to look for. So that's one thing. We know we only know what we know, and we're pretty much, a little bit tunnel visioned by the fact that these are the organisms we know about. But this is getting better and better as we identify more organisms. And of course, there's also now new tools that don't rely on probes and primers, but directly sequence. And we'll talk a little bit more about those later. Okay, so we're going to give a brief overview of each of the major domains of microorganisms. And we're going to start with bacteria. And just to back up for a second, soil is an amazing substance. We think that it's the most diverse environment or ecosystem in the world. Sometimes people who work in sediments fight with soil scientists and say sediments are, but I think soil, because it has some of its pores filled with air and not all filled with water like sediment is, it has a little bit more heterogeneity in it. So in a gram of soil, we estimate, based on sequencing that's been done and extrapolations made from those sequencing, we have 20 to 40,000 so-called species in a gram of soil. Of course, it'll vary by soil, but tremendous diversity. Species. Now, there's a problem with this word species, because the concept of species is not very useful for microorganisms. And I don't know that it's so useful for plants and animals, but, like in animals, we say if you get a offspring that can reproduce from genetic exchange or sex between two individuals, then they are within the same species. But this really doesn't make sense for bacteria. In the olden days, we used to classify organisms based on phenotypic traits and then try to put them in categories of different species, and then they'd go and contradict the trait that you said was attributable to your organism, because bacteria are always evolving and figuring out how to do certain things that they may not have been able to do when you were doing the tests on them. So they're pretty plastic, and so these phenotypic traits were not a good way to classify them. And then when you look at sequencing as a way of classifying and differentiating different organisms, what you have is then a continuum and no clear, obvious way to designate what % similarity allows you to be in the same group of species. So we have this rule of thumb that is kind of used, although you're going to find lots of exceptions to it, but this rule of thumb that greater than 97% similarity in your sequences make you a member of the same OTU, Operational Taxonomic Unit. Now, there are organisms that are more similar, might be 99% similar, but are very, very different in their behavior, and sometimes they get classified as different species. So, as I said, it's not a hard, fast rule, but it's a good practice. Does this mean that then it doesn't make sense to give species names to organisms? And no, that's not true. People still are always finding organisms and culturing them on plates and getting them in pure culture, and then running a bunch of tests on them and describing them, and then giving them names, species names, with like Pseudomonas putida, just like with animals and plants. Okay, oops. Here's another issue, though, that you probably touched upon in your lab. Only 1 to 3 to 5%. I mean, we don't know the exact number, but of species in soil can be cultured, or OTUs can be cultured, can be grown on plates, because Petri plates are just not the answer for a lot of organisms in terms of how they want to live. And because of that, it's this problem that I just talked about before. There's a lot of unknown organisms out there. We only know the tip of the iceberg, but this makes it really exciting for those of you who want to go into soil microbiology. There's so much new area to move around in and be free and make discoveries. Okay, so only about 9,000, maybe it's 10,000 now, species have been described, and a lot of these are pathogens, although it's getting better. And I also want to say that you can culture some organisms. There's a lot you can't culture, but then maybe you can really work hard and go beyond Petri plates and come up with clever ways, like in the antibiotic paper that I posted for you to read on SmartSite. And maybe with clever ways you can culture organisms, but then there's going to be a lot that you're never going to be able to culture. They may not be viable ever. They may be hanging out as organisms, but cannot be brought back to life, or they may be dormant, and you may have to do something special to like revive them. So let's take a look at a sort of a generic bacterial cell. This is some gram-negative. I'm sorry, I don't know that it's a gram-negative, but it's a bacterium, and a rod is what I'm trying to say. And you can see that there's a lot of information about them. If you take a look at a cell, there's all this interesting stuff. There's flagella, and there's membranes, and there's cell wall, and there's nucleoid, and there's ribosomes, and there's also just the chemistry that's of the compounds that are making up these different aspects of the organism. And all of this is like really valuable information that we can use for studying organisms in nature. We already know about the nucleoid, that we use the sequences from that to be able to classify life and maybe go looking for organisms in nature. But also the cell wall varies among different groups of organisms, and the chemistry of that cell wall can be really useful for also figuring out who is there in the soil, but it's also very useful for evaluating the role of microbes in, for example, the carbon cycle. Also the cell membrane, we'll talk a little bit about that as one of the methods, looking at phospholipids. Another way to characterize organisms. People also look at the flagellum, and so it's a different way of looking at an organism. It's you can think about how it all works, and the physiology of the organism, but it also is all this rich information that can be tapped into to study organisms in nature. So we have different shapes of organisms. I think bacteria are pretty boring. Coccus rods dominate. Rule of thumb is a bacterial cell is about 1 micron wide. In soil, conditions are usually more extreme. There's a lot of organisms that are like starving. So this 1 micron rule is more based on sort of fat and sassy lab-grown bacteria, and they may be smaller and actually even less than 0.2 microns and slip through your filter if you're backpacking sometimes. But in general, we use 1 micron as a rule of thumb. And then there is a filamentous bacterium in a group called the actinomycetes, that may have filaments up to 15 microns long, but there's still going to be this diameter of, rule of thumb about a micron. And there's some other shapes as well, but they're not as common in soil. Spores. So spores are interesting special structures that not all bacteria have, but some do. And there's two major categories of spores. One are endospores and the other are conidia. There, in fact, has been an ancient spore found in amber that you can read about in the textbook. But endospores are found only in the genera Bacillus and Clostridium, where they have this process that they go through when they see that things are going to get tough. They kind of make a good guess that they better go into this dormant phase to get through this hard time. And in that case, the contents of the cell reorganize into this very, very resistant form, which is called an endospore. These endospores are what Clostridium make and that we're afraid of in canning. If we don't properly pasteurize the food we're making, they can sometimes hang out and do damage on you because they can be very toxic as human pathogens. The other major kind, and the endospores, the Bacillus and Clostridia, they're common organisms in very arid environments, in like deserts, but it's not the only way to survive. It's just one specialization. Conidia are asexual spores that actinomycetes have, and that are held on these fruiting bodies that grow out of the cell structures. And in that case, one cell or one individual is going to make multiple spores rather than one cell equals one spore. And conidia aren't as good as endospores for survival of extreme situations, but they also help quite a bit to if you're in a vegetative state as an actinomycete and you decide, I better check out and make a lot of children, copies of myself to endure this phase that's coming up. And then when times are good, these spores can germinate and create new individuals. There's some other strategies than just spores for surviving extreme conditions, and sometimes it's just some changes in the composition of a cell. They'll create more resistant cell walls, or shut down some of their activity, and so that they don't require much energy to survive. They may shrink in size, go dormant. So these are other ways of surviving. And again, I just want to underscore, soil is a pretty tough place to live, particularly in some parts of the world. And bacteria have come up with a lot of interesting ways to get through hard times. Okay, another important aspect of bacteria, and archaea as well, is their cell wall, or their envelope, that is surrounding the cytoplasmic membrane, the membrane enclosing the cell itself. So we have two major kinds of cell wall structures that are pretty easy to differentiate with staining and a microscope, and you'll do it, in fact, in the next lab. But please here look at the cytoplasmic membrane in these two groups of organisms, the gram-positive one, which is shown here above, and the so the peptidoglycan is the purple here, and in the gram-negative, the peptidoglycan is shown here as this very skinny little layer. So that's the main difference between gram-positive and gram-negative, is gram-positive has this very thick peptidoglycan layer that is what is stained and what you're looking at for retaining color when you look at it under a microscope, which is outside of the cytoplasmic membrane. And it's made of both organic materials and minerals that make it very, very strong. It makes the organism quite rigid and is able to survive some of the extremes in salt content and desiccation, maybe wet-dry cycles as well, that I think of this organism kind of as having a big castle wall that sort of just protects it, and it's sort of safely underneath it, and it uses just this big physical structure to protect it against what's going on in the environment. The gram-negative is quite different. It has a very thin peptidoglycan layer. Again, you can see it outside of the cytoplasmic membrane, but then in addition, it has an additional membrane that is the outer membrane, and a gap in between the cytoplasmic membrane and the outer membrane called the paraplasmic space. And in this space, there's a lot of interesting things that can happen. The organism can detoxify bad chemicals out in the environment and prevent it from hurting them before it can go into the cell. Also, they can do some pre-digestion, I think kind of the saliva in our mouth where things start to digest as well. So it can partially transform compounds and then decide to take them up across the cytoplasmic membrane into the cell. And these organisms are more likely going to be plastic, engaged, respond to conditions in the environment, maybe modify them, which is a different strategy from the gram-positives. Okay, another interesting thing to think about is look at the chemistry that how different it is between these two groups of organisms. The gram-positives have all this peptidoglycan. They don't have this outer membrane, in contrast to the gram-negative. And so just think of these organisms now as feedstocks for soil organic matter, as precursors to carbon that's going to enter into the soil, because microbes are very important sources of carbon to the soil. And you can see that chemically it's going to be quite different. Like, what are the kinds of compounds that are going to be coming from the dead bacteria from a gram-positive versus a gram-negative, and it's going to influence the carbon cycling in the system. Okay, flagella are another structure that bacteria have, and that differentiates them, and we've got all different kinds of things, some of them coming out of just one side of the organism, some bundles of flagella or a single flagellum. And we generally think that flagella are used for motility, for movement. And it's interesting to think about soil in that soil movement is going to be much more limited than in an aquatic system. And movement's going to be most common, or the greatest amount of movement will happen when the soil is wetter, where there's pores between soil particles are going to be filled with water and organisms can move across them. But flagella still seem to be pretty important in soil, and it's probably because they are important for very short distances. And seems to be that organisms that live in the rhizosphere that are able to move with the plant root as it starts to grow through the soil, maybe bacteria with flagella are better at sort of moving along with the root as it moves through the soil, if they're not attached to the root itself. And also, there seems to be evidence that flagella are possessed by organisms that are good at using insoluble compounds like oil, like in oil spills. Some organisms that are good at breaking down oil are flagellated, and it seems to maybe help them drill into these solid phases of compounds. Okay, DNA, we've talked about this already, DNA sequences for the small subunit ribosome. And in bacteria, DNA is found indeed in the chromosome. The bacteria don't have true nucleuses, but they have chromosomes in a nuclear, they have chromosomes that are not in this nuclear envelope. But there's also DNA found in plasmids. So plasmids are circular DNA molecules that exist outside the chromosome, independent of them, that carry a whole bunch of different genes. What's interesting about plasmids is they're much more likely to move around among different organisms, and therefore they are a vector for genetic exchange through the process of conjugation. Also, plasmids sometimes are released into soil when organisms die, and are sometimes picked up by other organisms. So plasmids carry a lot of interesting, useful, and frightening genes. Antibiotic resistance genes often are found on plasmids, and in fact, plasmids are why antibiotic resistance seems to be able to spread so quickly among different groups of organisms. Also, resistance to heavy metals are carried on plasmids. Some biodegradation genes are carried on plasmids. Nitrogen fixation genes, a lot of them are carried on plasmids. And so you can find these traits. For example, biodegradation. There's a story in the Midwest about, I think it was some kind of herbicide that was starting to be used like 30 years ago, and there were very few bacteria in the soil that were able to biodegrade this herbicide, and so people would put it on and it would last a while and kill the weeds and be fine. But over time, what happened was that the efficacy, the usefulness of that herbicide became shorter and shorter and shorter, that they would put it on and its ability to kill weeds would leave and would be gone in like a couple of weeks. And so they would have to actually add more and spray again, and it would degrade really fast. And what they found was that the genes for that allowed the biodegradation of this particular herbicide that was originally on, maybe just one kind of organism in the soil, had moved via plasmids to a variety of different kinds of organisms in the soil, and suddenly there were, a number of different types of organisms that were biodegrading this herbicide and jumping on it as soon as it was applied, and so again carried through plasmids. So bacteria have very broad metabolic capabilities and use a huge variety of organic compounds, even ones that we call pollutants, as sources of energy and carbon. So this is including compounds that we as humans make. They also use a variety of inorganic compounds as sources of energy, use different electron acceptors, and can be autotrophs and heterotrophs. So let's look at these definitions here, the terminologies to distinguish organisms based on carbon and energy. So all organisms need a carbon source, and they need an energy source or an electron donor. They also need an electron acceptor, which we'll get into here, but is not shown on this table. So there's different, people use different these words in different ways, but in this class I want to be pretty strict. We classify organisms on where they get their carbon as autotrophs, who get their carbon from CO2, from the atmosphere. Auto, I don't quite get it, but auto, they get it by themselves, I don't know. And hetero, other, which they get their carbon from organic compounds. So we have autotrophs and heterotrophs that refer to the carbon source. And then in terms of the energy source, there's organisms that get their energy from inorganic compounds, lithotrophs, rock eaters, litho, inorganic compounds, light, phototrophs, and organic compounds, organotrophs. And then you put the two words together to do the proper description of an organism. So a photoautotroph gets its energy from light and its carbon from CO2. A phototroph is like a plant or a cyanobacteria among microbes. And then we have organoheterotrophs, who get their carbon and energy both from organic compounds. And that would be like humans, and it would be like a whole lot of organisms in soil. There's a tremendous talent in soil in organoheterotrophy. All right, so other terminologies refer to the electron acceptor. A lot of organisms do use oxygen as their electron acceptor. We humans do. And many bacteria and fungi and archaea in the soil also use oxygen, but they don't have to. So oxygen users are called aerobes, but those that do not require oxygen are referred to as anaerobes. And there we have two kinds: the obligate anaerobes that grow only in the absence of oxygen. They cannot have oxygen. They can't tolerate oxygen conditions, because, as we'll learn a little bit later, an aerobic environment is an extremely toxic environment, and you have to have a lot of tools to be able to deal with detoxifying in this presence of this oxygen. So there's anaerobes, but there's also facultative anaerobes. So these are organisms that are very versatile. If oxygen is present, indeed they will use it as an electron acceptor, but if not, they'll use whatever else is out there in the environment for an electron acceptor. So this can include nitrate, sulfate. There's a whole bunch of different compounds, and believe me, we will get into this because it's very important for biogeochemical cycling and for soil. And also in terms of the aerobes, there are organisms that can't live at atmospheric oxygen concentrations. They only live at low oxygen concentrations, and they are microaerophiles. There are a lot of these in soil because soil is filled with gradients of all these different compounds that are there for microbes to eat. And oxygen, also you'll see gradients from the atmosphere, and big pores into small pores. There'll be like different concentrations along these trajectories, or along these transects of oxygen. We'll get into that more later too, and it will make more sense. We also have these terms that refer to whether you can use really high substrate concentrations. You can go to those all-you-can-eat places and really pig out. So eutrophs are very good at monopolizing high substrate concentrations, but there's also oligotrophs that are organisms that can only use low substrate concentrations. Substrate here refers to a compound that an organism can use as a carbon and energy source. So oligotrophs we often miss them entirely, because when we enrich for organisms, we often offer them up like high concentrations of, say, glucose or cellulose or whatever it is we want to enrich them on, and maybe oxygen too. Well, we might put them in a shaking Erlenmeyer flask, and so they're just like bombarded with all these high concentrations and rich amounts of food, and they can't handle it. They die. They must have low concentrations. So you need much clever ways to enrich them and provide them with conditions that they'll be able to survive at. And we've missed a lot of organisms in soil because we tend not to pay attention to the fact that there are these differences in concentrations that organisms can live at. Okay, so that's an overview of the groups of organisms that we find in soil, and these terminologies apply to not just bacteria, but to all organisms in life. Okay, and classification, well, we already know that we use phylogenetic approaches to classify bacteria, and it's quite good for that, but I don't want us to forget that there's all these important phenotypic traits that used to be what we used to classify organisms when I was a kid. That's what we did, and it was a pain. But these are really important aspects, and we already have talked about some of these in the slides before, but your cellular and chemical characteristics and what you eat, and your antigenic characteristics. People used to use antibody approaches to identify organisms in soil. Before we had the molecular tools, genetic we already know, and then also just these traits of what they do, you know, whether they're a pathogen or what they do in nature, their ecological characteristics. So please, you know, remember that all of this information that we already have is extremely important. We often are going to be more interested in this information about organisms than their sequence of their small subunit ribosome. We want to know more like what they do, who they are, you know, what they do, their deeds, rather than who they are, rather than their name, that you might get from small subunit ribosomal sequencing. So all of this information is important. The groups that we get, though, out of the phylogenetic characterizations are shown here, and I've given a list of some of the most important soil organisms that and how they group. Again, this is just Wose's tree that's shown sort of in a different way, showing these different relationships. And there's no way I want you to like memorize these groups, and we're not going to spend a lot of time on names of organisms throughout this course. You can go and get plenty of that in other courses. But here are these major groups and some of the examples of organisms that fall within them. We have purple or proteobacteria, and within the proteobacteria here, there's all these different subgroups as well. And that's where we find mitochondria. The ancestor of the mitochondria is a proteobacteria. But lots of exciting organisms here, organisms uh, but that's again another really important group. And then we have cyanobacteria. We're going to touch base with them in a following slide because they're really important in some soils. And then we have spirochetes and green sulfurs that aren't so common in soil. But we also have bacteroids and flavobacteria, which are some of the organisms that break down complex organic matter like cellulose in this group. And there's planktomyces, which you might find in compost heaps and hot springs and also other places, and then chlamydia. We also have these incredible radio-resistant micrococci, these Deinococcus that have been found in like uranium spills up at the Pacific Northwest Battelle lab, where we, you know, this weapons research has led to creating all these horrible wastes that are like buried in the ground, and they've found some incredible organisms that live and transform like uranium in those wastes. And then green non-sulfur that again are not so common in soil. But this is just an overview of some of the main soil organisms. But as I said, we're not going to go in and memorize these. Okay, so just want to point out these two groups. The actinomycetes are the filamentous gram-positive organisms that make asexual spores. Look at, I'm just resonating with some of the information that we've already talked about before. You also have a plate that is enriched for the actinomycetes in your lab. You've given them glycerol, I think, which they tend to be able to break down better than other kinds of bacteria. They're going to have this fuzzy kind of appearance on the Petri plates, which is related to these spores, these asexual spores, these conidia that they're going to like grow on the colonies. And you should be able to look at these under the microscope, and it should be nice. They produce antibiotics. I always like to say the only soil microbiologist who ever will win the Nobel Prize, Waxman, got the Nobel Prize because he found antibiotics in actinomycetes. And of course they are very important for human health, and I hope they continue to be important and we continue to find new ones. Also, actinomycetes contribute to that smell, that characteristic smell when it first rains, particularly when it hasn't been raining a lot. There's this compound called geosmin, which is produced by streptomyces, that is released when it first rains, and you get that wonderful smell of the earth. And actinomycetes are good at breaking down complex organic matter. And here's some pictures of conidia of actinomycetes, just showing you that they're way more attractive looking than those boring little rods and cocci that most bacteria have. Here you can see these conidia being held and then will be released on these fruiting bodies that are coming out of these actinomycete cells. Okay, cyanobacteria are another really interesting group. They're photoautotrophs, and they are bacteria. They're formerly known as the blue-green algae. And they are really interesting organisms because they get their carbon and in many cases their nitrogen from the atmosphere. So think about that. They can actually be there on a rock and eke out a living because they get two of their primary nutrients from the atmosphere. And then they get other elements out of the rocks. And so they're really important in arid environments. They're present throughout most soils, but they tend to play a bigger role in arid environments, and that's because plants, those greedy things, haven't crowded out everybody else by creating shade, because plants grow up and harvest the light and engage in photosynthesis and end up leaving not a lot of the soil open for organisms that would directly colonize the soil. However, in very arid environments, cyanobacteria are actually really important sources of carbon and nitrogen. And like for the budget of that particular ecosystem, the flow of carbon and nitrogen are going to originate in cyanobacteria. So they're important primary producers, even though they aren't going to produce that much. And also cyanobacteria are really important in colonizing new soils, like think of a lava flow. Some of the first organisms to jump in there and start to create life, they're like the pioneers, the ones that first come in, are the cyanobacteria because they can bring carbon and nitrogen in out of the atmosphere. There's really interesting videos you can see on YouTube showing how quickly these organisms respond to rain in arid environments. They have such short windows when they can be biologically active, so they're very fast when the first raindrops hit. These dry, crusty things just come to life and just start like sucking carbon and nitrogen out of the air. And as fast as they can, for as long as they can, before things shut down again and they dry up. And we have these crusts, cryptobiotic or cryptogamic crusts in these desert environments that are very fragile. And so I show you this picture of a boot. And also people on road bikes sometimes like break these apart because it takes a really long time for them to get established. Cyanobacteria aren't the only members. There also will be fungi and maybe green algae. And lichen, which is a symbiosis between a cyanobacteria and a fungus, are also there as well. So really interesting kind of bacteria in soil in special conditions. Okay, the second domain that we're going to talk about are archaea, and I did forget to mention that the discovery of archaea is a major domain, and research on archaea in soil is very much in its infancy. And we didn't really ever even talk about them when I started out in science. So a lot of the information that you see about soil microbiology is not going to mention archaea at all, but you can just kind of assume whenever we're talking about bacteria, you should perhaps think bacteria and archaea. A lot of traits are shared by these two groups of organisms, even though we don't know so much about archaea yet. But there are a lot of similarities. So a lot of what I said before also applies to archaea. But let's look at what makes them different. So we know based on the small subunit ribosome that they're distinctly different from bacteria. They're their own separate domain. And they also differ from bacteria in their lipids and their cell wall components. They don't have peptidoglycan. In their lipids as well, they're quite different. Archaea are famous for being found in extreme environments of high salinity and temperature, acidity inside guts of organisms, but they're really common also in soil and sediment. And in fact, a fascinating discovery just 10 or 12 years ago was that nitrifiers, organisms that convert ammonium to nitrite, that we used to think were only this narrow group of bacteria, also include archaea as types of organisms that can carry out that process. And what does that mean? Because we know so little about what archaea like and what they need to make them tick. And if they're major nitrifiers in soil, there may be things that we're completely missing about that process. So stay tuned. Here's a table that is comparing bacteria and archaea, and again, look at the differences. There's differences in their lipids, ester versus ether-linked, the peptidoglycan or not. There's also different responses to different antibiotics or inhibitors, and I just wanted to sort of step back here and take stock of that, of why are these differences important to like soil microbiologists. And one reason is like these differences in lipids. When we get into methods, we'll look at this a little bit more, but we can use these lipids to differentiate major groups of organisms in soil, like to compare bacteria versus archaea, like if we don't want to do sequencing, because we don't always want to do sequencing. There's other methods, and sometimes these methods are easier or cheaper or don't dump as much data on your head as the sequencing molecular tools. So these lipids, we tap into those to help differentiate major groups. PLFA analysis is a method that looks at that. Also, the cell wall differences are important. We used to develop antibodies to differentiate organisms. I don't think we really do that anymore. And I already talked about how that cell wall difference can affect the carbon cycle. So that's very important also in the difference between archaea and bacteria. And the fact that there's differences in these inhibitors, we can use inhibitors to try to tease out in the soil what is the role of bacteria versus what is the role of archaea in carrying out different kinds of processes. And in fact, in your lab that's looking at respiration in Soil Science 111, we are going to use some inhibitors to try to separate out the importance of fungi versus bacteria, because they have different sensitivities to different inhibitors, to look at the differences of them and their contribution to respiration in the soil. So again, this is just sort of echoing my earlier picture of all this information that was found in an organism and how you can tap into that information and use it to study microbial ecology. And this is kind of following up on that, again, looking at some of these differences and how we can use them to answer these really interesting questions about nature. Okay, so back to the archaea. So far, what we know based on sequencing is that there's several major groups of organisms, including the Crenarchaeota, the Euryarchaeota, the Chlorarchaeota, and the Nanoarchaeota. The Chlorarchaeota, this group haven't been cultured. We know almost nothing about them. And the Nano, we only have recently found out about them, and I think there's just one species, or maybe there's more now, but they're pretty unknown. So the Crenarchaeota and the Euryarchaeota are the main organisms in soil. kind of archaea in soil, and you can see here's their own a little tree that relates to what I'm talking about here. The woes tree, there's the archaea, there's the bacteria, there's the eukarya again, just so that you can get your bearings there. And among them are nitrifiers, methanogens, some other halophiles. And we can also look at them not just based on their sequencing, but by their physiology, right? So these are physiological groups, based on what they eat, or what they can survive, the kinds of conditions they can survive, so that are important and important to soil, to certain extreme soil conditions. So methanogens produce methane, and there's halophiles, extreme halophiles that can live under very high salt conditions, and extreme thermophiles that we also find in compost heaps and around hot springs and in different environments. But anyway, it's a different way, a more functional way of grouping the archaea, and this is the phylogenetic way of grouping them. Okay, sorry I don't have much more to say about archaea. I'm sure that we're going to just be finding out so much new information about them over the next 10, 20 years that's going to change, completely change how I would talk about them in Soil Science 111. Anyway, the third big domain are the fungi, are the eukarya, and in the eukarya are the fungi, who are another really important group of soil microorganisms. Similarly to bacteria, only a small percentage have been described yet. We think there's many, many more species out there. They are larger than bacteria, cell diameters of 2 to 30 microns. A lot of fungi are filamentous. Not all fungi are filamentous, but a lot of them are. And the way that these structures that fungi have are a hypha. An individual hypha is made, it directly corresponds to a cell, but then the hyphae bundle together and make up mycelia, which you start to be able to see with your eye, these bundles of hyphae, and then the mycelia together form thalluses, and thalluses are these larger bodies, these things like mushrooms that are made up of all these bundles of hyphae that we see above ground that are important for dissemination of spores. Okay, so with fungi we have this problem of what is an individual. We talked about that in class a little bit. When you do your plates, your Petri plates, enriching for fungi, you're going to find a bunch of colonies, and does each colony come from a separate individual organism? Probably not. You may have broken up a long hypha into fragments, and each one of those fragments could lead to a separate colony, making you then think like, okay, what does this number, this density that I'll base on plate counts, what does that mean for the soil? It's not a very good way of quantifying fungi in soil. And in fact, all of those colonies could come from one organism. We now know that the largest organism in the world is not a blue whale, it's a fungus. Fungi can one individual can colonize an enormous area of land, 1,500 acres. I think there's areas even bigger than this. Thousands of tons of biomass. You can go in and sample those hyphae like across all these acres and see that they're genetically identical, that they're the same individual, even though they are widespread over such a big area. So microbiology makes you question a lot of these things that we sort of take for granted. What is an individual? What is a species? It's good to be challenged. And in terms of biomass, they're often the biggest biomass in the soil is fungi. Not so much in agriculture. Agriculture is not a friend of fungi. A lot of agriculture isn't, especially if there's tillage and using pesticides. The tillage is constantly going to be like breaking up these continuous hyphae that start to form and disrupting them, and then they kind of have to start up again. So it's always disturbing them, and so bacteria tend to be more dominant than fungi in a lot of ag soils, and perhaps there's services that we're missing out on because of that. I think the other thing I want to point out is a mushroom is an above ground component of what is the more important part of fungi, which is all these hyphae growing below ground. So again, like a plant, there's a lot that's going on below ground that's equally, or in the case of fungi, more important. These fruiting bodies are mechanisms for the fungus to get its spores out in the world, to get its progeny, out of the home, kick their kid out of the house. Imagine if they release their spores right around where they are below ground. They would colonize and compete with them in the same location. And this allows them to pop up this structure that's filled with spores hanging from it, and then you've got like birds or eating them and carrying them off, or raindrops or wind, or all these different kinds of phenomena that then help move and disseminate these spores out in the world and get them into new locations and colonize new areas. So that's the relationship between mushrooms and reproduction. And there's some great videos here to watch of fungi. So most fungi are aerobic organoheterotrophs, but not all of them are, but most of them are. And they're very good at degrading complex organic matter. They can survive at low pH, probably because of their cell walls. They have resistant materials in them, sometimes chitin, and it could be melanins or tannins, these very complex organic compounds that help them survive predation as well as environmental conditions that are difficult. And the other amazing thing about fungi is that they are really good at staying metabolically active at very low moisture contents. And we will look at this a little bit more when we start to look at the soil physical properties of soil and how it relates to microbiology. But in a nutshell, what happens is that most organisms in soil, most microorganisms, have to live in water films because they are essentially aquatic organisms. So they're sort of stuck inside these, what in a dry soil can be a very, very thin layer of water. What fungi can do is, of course, they are also in those water films, but they can send a hyphae across an air-filled gap, an air-filled pore, over to another spot in the soil that has water in it and may have nutrients in it, and translocate back the nitrogen or the carbon or maybe even the water that's in that area back to like the mothership, the original organism from which sent that hyphae out. So they can kind of eke out a living across, by capitalizing on these little pockets of water and nutrients that are what happens when a soil gets dry and the water film breaks up into these little isolated pockets. So very good at that. This is a fantastic website. I don't know how active it still is, but it's a great place to get introduced to the diversity of fungi. Okay, fungi have three kinds of structures. Filaments, hyphae are the most important structure, but there's also yeasts. Don't forget that yeasts are also fungi. and they look more like bacteria. Actinomycetes look like fungi, right? And they're really, really important in certain aspects of soil. They tend to live on the surface of plants, leaves a lot, and in flowers, but they're also present in soil. And we also have this weird chytrid kind of fungus as well that I don't think is so important in soil, except some pathogens. So we also have these septa. So here's like a filament of a fungus, a hypha of a fungus, and there's different kinds of septa that are found that are breaking up these hyphal cells. So there's some that have no septa at all, they're aseptate, but there's also others that have these different kinds of structures. Here's what is called an ascomycete, and this is a basidiomycete, that have these different kinds of little portals, I guess, that let things move between individual cells, because of course fungi do have this ability to translocate material across different cells along the hyphae. So that's something that you might see under a microscope, and an actinomycete won't show these septa. They will just show individual cells, or differentiable cells making up the filament. And then this picture here shows you the hyphae, and then this fruiting body with the conidia that is forming off of this original hyphae. So another kind of structure that you might see, and that you might see when you look at the plates in class in the next lab. And then there's two major kinds of spores. Asexual spores and sexual spores. We already have talked about conidia. You know that some fungi have them, and remember that actinomycetes have them as well. But also fungi have sexual spores that are formed from the exchange of genetic material between two individuals. And these major kinds of sexual spores are zygospores, ascospores, basidiospores, that just happen to be the basis of taxonomy of fungi. And you can see that here. Our major groups of organisms are ascomycetes. There's a picture here shown of one. Basidiomycetes, which are a lot of the more famous mushrooms and shelf fungi, are in this group. Zygomycetes, which are like bread molds and less spectacular organisms in their appearance, in terms of their fruiting bodies, but are very important in decomposition. And oomycetes, which tend to be more in aquatic systems. And we also have these deuteromycetes, which are the fungi imperfecti. This group used to be more important when we didn't have sequencing information to be able to categorize them into these other groups. But they still exist. It's kind of like where we put organisms that we haven't seen their sexual spores. So these classifications based on the sexual spores also are supported by the sequencing groupings as well. So the small subunit ribosome and the other regions that are used to sequence fungi are compatible with these groupings based on the sexual spores. I wanted to say one thing is that there are other structures that fungi have to survive stress, and these include particularly sclerotia, but also maybe rhizomorphs, which some fungi have, these other specialized forms of structures to deal with the harsh conditions of soil. Okay, another major group of organisms are slime molds, and slime molds also are eukaryotes, and they fall out actually in two major groups in the tree of life. You can go back and look at Wose's tree and see where they are. They're kind of in between the fungi and the branch where the eukaryotes come out. And they are fascinating organisms, and I love them. They're beautiful and they're interesting because they change a lot. And they have, and I really recommend that you go look at some of these videos of them. And you may have noticed them. You're walking along and you see this, maybe these beautiful, beautiful, like golden globes, or, or colorful little blobs of things in woods. Or you might see something that looks like vomit, like this big, looks like someone got drunk, and it's there in a, maybe around the base of a decaying tree, or often you can see them in flower pots. They may look like scrambled eggs, like this here. And that's the early stage of the slime molds, because they start out, they're spores that germinate, and they create, they aggregate. So the individuals that germinate are actually very fungus-like. Well, actually, they're not fungus-like yet. They're more like protozoa-like, and they will come together in a big mass, this collective mass of many, many individuals who have germinated, and some people call them like a wolf pack. They you find them in areas where there's a lot of nutrients, something like fresh mulch or decaying tree, and they will roam over and eat the bacteria that are there, eating the these nutrients that have become available. And I mean, I think it must be terrifying to see this, what's called a pseudoplasmodium, coming at you, which is made up of all these individuals that are going to engulf you in a minute. And then they go into the second phase where they become more fungus-like, and they start to fruit and create these fruiting bodies, where they will no longer be active, and motile, but they'll take root somewhere and reorganize themselves into this like stalk, and then create spores that are held on this fruiting body, and then they sort of turn to powder and it blows off. And if you go back and look at that, like vomit that you might have seen a couple of days before, you'll see now that it's like all dried up and powdery, and maybe even has disappeared. So really, really interesting organisms, predators of bacteria and perhaps fungi as well. And then we have the protozoa. So the protozoa are also eukaryotes, and they're found in that, again, scattered throughout that area between where the plants, animals, and fungi fall out in Wose's tree, and the branching place where the eukaryotes branch off from the archaea and the bacteria. We used to think they were all very related, but they are actually quite different. I've starred the most important ones in soil, which are the flagellates, pictures shown down here, the two kinds, and then the amoeba, which are really important in soil, both the naked amoeba and the testate amoeba, and the ciliates, shown down here. They're on the order of 10 to 100 microns long. There's maybe 30,000 species. A lot of them are phagotrophs, meaning that they engulf their prey. So they're really important predators of bacteria, archaea, and sometimes fungi. Some of them are organoheterotrophs, or have some ability to eat organic matter directly, but a lot of them are predators. They differ in their importance in soil. I think the most important ones in soil are the flagellates, which are very small, and can live in these small films of water that happen, that is the condition of water in soil a lot of the year. In contrast to the ciliates, which are quite a bit larger and kind of rigid. And so they're going to have a hard time when the soil gets dry, because they're not going to be able to move around, and they're quickly going to become too big for the amount of water that's there. And then the amoeba are particularly adaptive to soil conditions because they can reconfigure themselves. I always think of water films going from a thick water film, and the soil dries out and the water film gets thinner and thinner and thinner, and it can get as thin as like a couple of microns or even less. And I can just see that amoeba just hunkering down, stretching itself out, living in that narrow water film. This may be my imagination, but they have a physical structure that makes them better adapted for the low conditions of low water conditions of soil. And then we have other organisms in soil as well that we're not going to spend too much time on, and these include what we call the micro and macrofauna, and are nematodes and microarthropods, including macroarthropods, which are the larvae of a number of insects that may have some stage of their life in soil, earthworms, we talked about shredders, and then of course there's mammals and some birds that spend some time as well in soil. And this gives an overview of functions of soil organisms, so photosynthesizers, decomposers, mutualists, but then also root feeders, so that's one role of some of the larger organisms in soil, the fauna, but also bacterial feeders, fungal feeders, these predators. And remember, the front line in the food web are the shredders that are really important for breaking down organic matter and inoculating it, and providing opportunities for a lot of other organisms to deal with their byproducts. And then we have the other organisms. And I'm sorry, there's a lot of really interesting organisms here that we're just not going to spend much time on, except perhaps to look a little bit at their function, like this releasing nitrogen from bacteria during predation. But anyway, okay. And these are some great pictures, some of them nightmarish, of some of these organisms in soil. And I think we'll have a microscope to be able to look at some of these in compost at the next lab. If you're lucky, you'll see a pseudoscorpion or these mites, and collembola are the little white dots that sort of are pop, 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 jumping all around in compost. And nematodes, tremendous diversity to nematodes as well. Okay, and then we have viruses, so we don't want to forget about them. They're not strictly organisms, but they're, as you know, acellular, intracellular inhabitants of life forms, obligate parasites. They're not on the tree because they don't have small subunit ribosomal RNA. It's a rule, and I think it's interesting to think about them both as genetic elements that contain, there are DNA and RNA viruses that replicate in cells, but they also have an extracellular state. And then there also can be considered obligate intracellular parasites whose DNA or RNA is in proteins encoded by their genomes. Again, we're not going to spend a lot of time on them. They're very small, but very present in soil, 20 to 300 nanometer diameter. We classify them based on their genetic information, their morphology, their size, and who their hosts are. And here's some of the major families of them for your viewing interest, but not to worry about too much in this course. But let's think a little bit about their ecological importance. They are important for transduction, for moving genetic material between different hosts, so could be very important in that process. Also, they're parasites, so they can be very important in controlling bacterial populations. Bacteriophages are what we call viruses that live in bacteria, and in fact we can see like higher numbers of rhizobium bacteriophages in fields that have legumes in them, because there's a lot more rhizobia there. So you can see this correspondence between the presence of these viruses and the presence of the organisms that are their hosts. And also the viral shunt is when the host is lysed, or broken open to release the viruses, there's also going to be release of nutrients that will go into the soil. So maybe viruses also play a role in liberating the nutrients that are tied up in microorganisms. And there's human health issues associated with viruses, as well as with some of the pathogenic bacteria. And we find viruses in landfills and septic tank drain fields and effluents, and where sewage sludge is applied to soil. It's been a concern in the past. Around feedlots, cemeteries are great sources of pathogens and viruses. And the thing about soil is viruses can really survive well in soil. They can sorb to, and sorb, that means stick to the outside, not absorb, but adsorb. So through chemical interactions, they can be held to clay particles or organic matter, which helps them to survive over time in soil. And they also may form resistant structures. And we can count viruses the same way that we count bacteria on Petri plates, by growing up a lawn of bacteria, like this continuous sort of just film of bacteria on a Petri plate. And then you take your soil and dilute it in the same way that you did for your bacteria. And everywhere there's a virus that in your diluted soil suspension that you pipette on this lawn of bacteria, everywhere there's a virus, you're going to get a clearing, a zone of clearing. That's what these little dark dots are in that lawn of bacteria, that's going to show that there is a virus there. So you can use it to enumerate viruses, or count how many viruses you have in your soil. Okay, I'm going to stop here and take a break, and then move into part 2 of this lecture. Thanks. I hope you're still here with me.